Thank you, Melanie, and I just want to thank the organizers for the opportunity to share my work, and I'm really excited to be part of this community. I started my lab just 2 months ago, so today I'm going to share some work that came from the end of my postdoc at Harvard. So my lab is focused on understanding how viral proteins and antibodies evolve so that we can predict the emergence of new viral variants and the efficacy of the corresponding immune response. And this is, of course, underscored by the ongoing SARS-CoV-2 pandemic, where over the past 2 years we've seen multiple variants of concern emerge, and we've rushed to test whether our current vaccines and therapeutics are still effective. But instead, if we understood how these variants emerge, we might be able to develop therapeutics that are effective against emerging variants and refractory to the development of further resistance. And the likelihood that some new variant emerges and increases in frequency is related to its fitness or its ability to replicate. And this fitness is determined by changes in the viral genome. So if we just consider the diversity along the SARS-CoV-2 genome for the Omicron BA.1 variant, we see that there's diversity across the genome. But there's really considerable diversity in the region encoding for the spike protein. And so often to understand why some particular variant increases in frequency, it can be useful to focus on individual viral proteins. And so we can define protein fitness as the ability of that protein to perform functions that promote viral growth. So for the spike protein, it needs to bind the host ACE2 receptor, it has to mediate fusion with the host cell membrane, and as the major antigenic target of SARS-CoV-2, of course, it has to evade neutralizing antibodies. And then to understand why some particular variant has improved fitness, we need to know how the mutations that it has change these properties. And this sort of thing has been done for many proteins beyond viral proteins by mapping what we call protein fitness landscapes. So these landscapes define the relationship between protein genotype and fitness. So for example, if we just consider some wild type protein that just consists of 3 amino acids, that protein can evolve by mutating each of those 3 amino acids, and those mutations might change its fitness. And then we define the fitness landscape of this protein by all possible genotypes it has access to and their corresponding fitnesses. And this information is really useful for understanding the likelihood of specific evolutionary pathways. So for example, if we assume that selection pressure is strong, then mutations will be favored if they improve fitness. And so we might expect certain evolutionary pathways to be more likely than others. And these landscapes have largely been mapped by advances in sequencing technologies. So several groups have developed evolutionary inferences that leverage existing genomic data to infer the fitness of mutations based on their frequencies, where mutations that are present at high frequencies are assumed to enhance fitness. And then other groups have used experimental measurements where they actually construct these variants in the lab and then use sequencing-based technologies to measure their fitnesses in high throughput. And so again, this has been done for many proteins beyond just viral proteins. And in general, there are 2 key insights that we are observing quite frequently. The first is that the fitness effects of mutations can vary considerably across different selection conditions. So for example, if we consider some viral landscape in the absence and presence of some monoclonal antibody, the mutations that it has might be really different in these 2 different selection environments. And so some mutational pathways might be accessible in the absence of the monoclonal antibody that are no longer accessible in the presence of that antibody. The second key feature is that these fitness effects also depend on the presence of other mutations. So for example, mutation at this first position might be neutral if it occurs in the wild type background, but might become beneficial if it occurs after mutation at this third position. And so these sorts of non-additivities are known as epistasis, and they make evolution pathway dependent, meaning that the order in which these mutations occur matters a lot. And together, this really means that the fitness effects of mutations are highly context dependent. So they depend on the selection pressure, and they also depend on what other mutations are already there. And this context dependence is really important in the context of viral proteins, because these proteins are constantly experiencing new selection conditions, and they're acquiring these mutations really rapidly, meaning that there are numerous opportunities for these mutations to interact epistatically. And so we've used several approaches to try to map these context dependencies for different viral proteins and different antibodies. And what I'll talk about today is work that we did trying to understand the evolution of the BA1 variant. And so we started this project about a year ago, right when BA1 emerged. And as evolutionary biologists, we were quite surprised that this variant emerged from the ancestral Wuhan variant that hadn't been widely circulating for many months, rather than from the widely circulating Delta variant. Not only that, this variant had many mutations relative to the other variants of concern that we had seen emerge in the recent past. And so, to understand why we see the emergence of this variant, again, we're just focusing on one protein, the spike protein, and in particular, the receptor binding domain. And we wanted to know how the mutations in the receptor binding domain are changing these different properties of the spike protein, and how this might constrain evolutionary paths to Omicron. And the way that we tackle this question is by mapping all possible evolutionary intermediates between the ancestral Wuhan and Omicron sequence. And so we generate all 2 to the 15 possible combinations of the 15 mutations that separate these sequences, and then we measure biophysical properties that we know to be important for the evolution of spike protein. So we measure binding affinity to ACE2 and also to neutralizing antibodies. And so again, there are 15 mutations in the RBD of the spike protein. They're distributed across the RBD. We use some combinatorial methods to generate this library of all possible evolutionary intermediates, which contains all 2 to the 15 combinations of these mutations, which is about 32,000 different sequences. And then we measure the affinity of those variants to ACE2 and to a panel of antibodies. And so to do that, we use a method called tight-seq. So this is a high throughput method where we can measure all of these binding affinities in a single experiment. And we do that by putting our RBD library in a plasmid, transforming it into yeast such that each yeast cell expresses many copies of that variant, but only of one particular variant. And then we label that yeast library with some fluorescent protein, so for example, ACE2. We then measure binding affinity using a combination of flow cytometry and sequencing, where we look at the fluorescence of our library, which I'm showing here with this green distribution. Variants that bind ACE2 really well will be in the upper end of this fluorescence distribution, and those that bind less well will be in the lower end. And we can determine which variants lie where by sorting this population into different bins and then sequencing those bins. And if we do this at multiple concentrations of ACE2, we can fit the mean bin that we observe each of those sequences in to the concentration of ACE2 and infer equilibrium binding affinities. And so we do this for all 32,000 variants, and these data are highly correlated across replicates. The error on these KD are about 0.1 log units, and they correlate quite well to isogenic measurements as well as solution-based measurements that have been made by many other labs. And so first I'm going to show you the binding affinity data to ACE2. So I'm going to show you affinity to ACE2 for all 32,000 evolutionary intermediates. And those are now displayed in this histogram, where on the x-axis I'm showing the minus log KD. So here a higher number is stronger binding affinity. And we see that the Omicron variant is among the best binding sequences in our library. It's almost a half log better of a binder than the Wuhan sequence. But really interestingly, we see that a substantial portion of our library has reduced binding affinity relative to Omicron, meaning that many combinations of these mutations actually reduce affinity despite Omicron having improved affinity. And when we parse this by the number of mutations that are present in those variants, we can look at those that have reduced affinity relative to the Wuhan variant, which is represented by this dotted line. And we see that those variants that have reduced affinity have some intermediate number of mutations, but that once they're highly mutated, there really aren't any variants that are reduced in affinity relative to Wuhan. And so we wanted to understand what's going on here. How are these 15 mutations interacting to produce a landscape like this? And so to do that, we look at the effect of each of these mutations. So now I'm showing you the change in binding affinity. So here, a positive number is an improvement in affinity, and a negative number is a reduction. And I'm showing you a violin representing the effect of a given mutation across about 15,000 different genetic backgrounds. And then I'm pulling out the effect of that mutation on the Wuhan background, so going from having no mutations to having that one mutation, and on the Omicron background, so going from having 14 mutations to all 15. And we see a couple interesting things. So first, there are some mutations that are largely neutral. The violins are centered at 0. There are some that are quite often deleterious. Almost the entire violin is below 0, meaning that on all genetic backgrounds, these mutations are deleterious. And then there are others that are quite beneficial, improving affinity by up to a couple orders of magnitude. And when we look at the mutations that have reduced affinity, we saw that these largely correspond to known antibody escape mutations. So there's a cost in ACE2 affinity to acquiring these antibody escape mutations. But when we look at the effects of these mutations on both the Omicron and Wuhan backgrounds, we see that in general, mutations have a deleterious or neutral effect on the Wuhan background, and then become beneficial in the Omicron background. And even these antibody escape mutations are quite deleterious on the Wuhan background, but then almost become neutral on the Omicron background. And so we knew that there was some compensatory epistasis in this landscape, and in particular, we thought that that might be with these highly beneficial mutations. And so we wanted to look at interactions between this set of 7 mutations to understand how this variant is maintaining affinity to ACE2 and also evading these antibodies. And so we do that just by sorting our data. So first we look at variants that don't have these beneficial mutations, but have either none, one, or all of the antibody escape mutations. And again, we see that if you acquire one or all of these escape mutations, your affinity decreases by up to 2 orders of magnitude. If you have one of these mutations, so 501 or 498, this reduction is reduced. So now your binding affinity is only being reduced by maybe 1 order of magnitude. And when you have both 498 and 501, this loss in binding affinity is completely abrogated. You can obtain all of these antibody escape mutations and maintain affinity to ACE2. And so this compensatory epistasis is enabling Omicron to be able to evade these antibodies. And so what does this mean for evolutionary paths? So because we have this combinatorially complete landscape, we can actually look at all possible evolutionary paths between these 2 sequences, but here I'm just showing you a random subset of 100 of those paths, and I'm coloring them by whether or not those compensatory mutations are present. So if none of them are there, they're blue. If one or the other, they're black, and they're red if both are there. And I know there's a lot going on here, but what I hope you can appreciate is that all of the paths that are flat or increasing are black or red, meaning that you have to have these compensatory mutations to be able to acquire these antibody escape mutations if you assume that some threshold of ACE2 affinity is important for maintaining infectivity. And so this begs the question of why Omicron has all of these mutations. Are all of them necessary for evading these antibodies? And we wanted to address this question, again, using our tight-seq method, but here, instead of measuring affinity to ACE2, we actually measure affinity to different monoclonal antibodies. And we chose 4 antibodies that bind different epitopes of the RBD. And perhaps not surprisingly, given that we chose antibodies that bind different epitopes, we found in general that the mutations that enable escape are distinct between these antibodies. So for this Eli Lilly antibody, we saw that this mutation K417N is the only mutation required to completely escape the antibody, which is represented here by non-binding. For this S309 antibody, we saw that there were no variants in the library that could escape this antibody, including Omicron, although there were variants in the library that had reduced affinity relative to Omicron. And then for these other 2 antibodies, we saw something more interesting, where multiple mutations are required for escape. For this Regeneron antibody, you need 2 mutations to completely escape it. Having either one individually doesn't really do much to binding affinity. And for this Eli Lilly antibody, again, having 2 mutations is required for escape, but having either one gives you some intermediate effect, where it reduces affinity by a couple orders of magnitude. But again, if we just look across these antibodies, the mutations that enable escape are distinct. And not surprisingly, they correspond to those that are in the footprint of the corresponding antibody. So the large effect mutations are in these footprints. And so we next wanted to know whether there were alternative pathways to escape. Are these large effect mutations the only way that you can escape these antibodies? And the way that we addressed this question was using a decision tree analysis, where we take our antibody escape data and we split it by mutation. And if that mutation can completely explain whether or not that genotype escapes the antibody, this branching process stops. If it can do better by adding another mutation, it continues. And so just as an example, this antibody, which variants can completely escape by acquiring one mutation, totally stops after splitting the data by this 417N mutation. For this Regeneron antibody, where we saw that 440 and 446 were required for escape, again, we see that in our decision tree analysis, where these two mutations lead to escape 100% of the time, which is represented by this red bar. But we also see that if you don't get mutation at site 440, but you get 375, you can escape this mutation about 80% of the time. And if you don't get 375, but you get 498, contingent on not getting 484, you can escape about 70% of the time. And so there are alternative pathways to escaping these antibodies by smaller effect mutations that are not in the footprints, but these escape pathways are epistatic, and they likely depend on the effects that these mutations have on other properties of this protein as well. And then for this other antibody, it's even more complicated. In general, though, if we just look at the effects of these mutations on ACE2 affinity and on affinity to these different antibodies, averaged across genetic backgrounds, we see that each of these mutations provides a functional advantage to BA1. So it is using distinct sets of mutations to escape these antibodies, and then an orthogonal pair to compensate for that loss in ACE2 affinity. And this is really rare, that every mutation in a protein has a functional advantage. And so it speaks to how Omicron may have evolved. And so thankfully, in most of us, when we get infected with COVID, it's an acute infection, meaning that when it replicates in our cells and mutates, those mutations don't have enough time to reach high enough frequencies to be passed to the next person because of these transmission bottlenecks. But in the context of a chronic infection, variants do have enough time to rise to high enough frequency where one or multiple mutations can be passed to the next person. And because our affinity data suggests that you really need a set of mutations to confer this fitness advantage, it is consistent with the widespread observation that Omicron is most likely to have emerged from a chronic infection. And so today I've shown you how this context dependence might shape the evolution of the Omicron variant. So we see that different mutations affect escape from different antibodies differentially, and the pathways to maintain ACE2 affinity and to escape these different antibodies are highly epistatic. And so this context dependency might make you think that predicting evolution is entirely hopeless, but I would argue that the fact that these context dependencies are defined by measurable biophysical properties means that this indeed is predictable, and we just need to be measuring the right things. And so today I've just talked about affinity. We've heard a lot of talks today about how there are many other properties that matter for these viral proteins. And so my lab is now developing high throughput methods to measure some of those different properties to integrate with this type of data. And so if any of that sounds cool to you, I am recruiting grad students and postdocs, but I also just want to thank the awesome team that I collaborated with on this work in Michael Desai's lab, so Elif Milana, Thomas Dupek, and Jeffrey Chang, and then our collaborators at the Hutch and Jesse Bloom's group. And all of this data is available online in the form of a browser. I encourage you to explore it if you're interested. I'm happy to take any questions.
Okay, go ahead.
Oh, thank you for the amazing talk. When somebody's infected with a quasi-species of coronavirus, has anybody looked at the prevalence of extremely rare variants that might represent the primordial strains? In regard to that, to what extent does coronavirus's amazing ability to recombine play into this process?
Yeah, so I think if someone maybe from Raul's group is here to comment on the quasi-species, they might be better able to. I haven't seen any like super low frequency variant sequencing data, so it's hard to measure. You need a method like CIRSEq to be able to do that, which is technically difficult. And yeah, I mean, recombination can do a lot of things for epistatic landscapes. So it can, of course, separate deleterious mutations from beneficial mutations, and that might make it more likely to see multiple beneficial mutations in a genome and the absence of deleterious mutations. Yeah.
Hi, great talk, and congrats on the new lab. But I guess two questions are like, one's past-facing, one's future-facing. So using your data, do you think you could maybe retrace the hypothetical evolution of like which came first? Is it the antibody escape mutations and then it gained the ACE2 binding so that it could transmit, or is it the other way around? And then in the future, do you think, could you adapt your technique to using just, let's say, the Omicron spike now, how it could evolve further given the current antibody landscape, like using human sera or something like that?
Yeah, thanks. Your first question was
about
great.
like, could you use the evolutionary data to figure out like, which came first?
So we can't really know unless we know the relative importance of the different selection pressures. And so, yeah, some recent data has come out that we're now integrating with ours, where there have been intermediates to Omicron observed in some HIV-infected patients, and so we can try to think about what those evolutionary intermediates are. If you assume that you need to maintain this affinity level to ACE2, then we think those compensatory mutations came first, but yeah, we really don't know. And yeah, future looking, the way that we're sort of exploring the landscape is combining this combinatorial approach with Jesse Bloom's approaches of deep mutational scanning. And so you can do that to kind of understand what the shape of epistasis is for these proteins, and then also what the local mutational landscapes are, and use that data in a predictive model. Yeah.
Thank you.
Thank you for the great talk and doing a great job on explaining all the complexities. I was wondering, kind of going more towards the One Health perspective, whether you can adapt your approach to looking at species barriers, for example, looking at bad ACE2 and how basically that is a difference instead of mutations that escape immune response, but rather allow the transmission between species.
Yeah, so we are looking at that, and yeah, Tyler Starr is also looking at that now in his group that he recently started at Utah. Yeah, so we're excited to do that with him.
Thank you.
Great talk. I would argue that it probably is not the monoclonal antibody pressure that is selecting for a lot of these. I can tell you as the developer of S309, it wasn't that widespread of a use, and so I would argue, especially now that we have no monoclonal antibodies that are currently approved for use due to changes in the variants, that looking at sort of polyclonal sera. My other comment is using yeast display, and I've had many conversations with Jesse about this as well. As the virus actually accumulates mutations, it's not only just the amino acid differences, but it's also the sugars. And so using yeast display, do you think that maybe we're not capturing everything? So you're capturing the major things, like 484 and all of those sort of mutations. But how are you thinking about sugars, which are the bane of our existence right now?
Yeah, so thank you for those questions. I come from a protein folding and proteostasis background, so I care a lot about those things, which is why I'm moving these technologies into human cells. Yeah, I think they matter a lot, and I'm doing that. To your first question about the monoclonal antibodies, so yeah, these are unrealistic selection pressures. The reason that we chose them was because you can't measure binding affinity to a polyclonal sera, and we just wanted to essentially look at the largest space possible, so we chose things that bind different footprints. Yeah.
Awesome, thanks. I guess I'm just really sensitive to the thought that it's monoclonals that are selecting for these
That's fair.
Being at Regeneron and then at Vir.
Yep.
So completely different target for the protease inhibitors. let's talk about nirmatrelvir of Paxlovid. In their drug fact sheet and in many literature papers, there are known drug-resistant mutations and known adaptive mutations. The drug-resistant mutations are largely growth defective. So it's possible that if we start looking for the adaptive mutations before the resistance, it's possible those adaptive mutations are necessary on the evolutionary scale timeline before we start seeing them. It could be worthwhile to use your approach to try to predict, with biochemical assays that are available and viral assays, what pattern we should be looking for within the the circulating populations to signal if there's going to be resistance emerging.
Yeah, definitely. I think, yeah, the hard thing is that like these compensatory mutations have no effect on the Wuhan background. So like we would have never predicted that they're beneficial mutations because they're really only beneficial in the context of the Omicron variant. But yeah, I'm definitely interested to look at that. Okay, thank you so much.
Well, thank you, Angela.